Diagnosis. This topic is taken from the chapter of biotechnology and its applications. So, first of all, you should understand what is molecular diagnosis. Do you know what is the meaning of diagnosis? Diagnosis is basically the identification of illness. Okay, recognition, right? When we take the disease and talk about its effective treatment, then we need to know how to treat this type of diseases, right? So early diagnosis is required to treat any type of diseases. First of all, we should identify the disease, which is very necessary, right? For example, when cancer is in third stage, right, first of all we should know the method of treatment of the cancer in the third stage. So earlier diagnosis is very necessary. Why early diagnosis is necessary? Because early diagnosis is very necessary because the chance of survival of the person is very high, right? With very more. Okay, early diagnosis is only possible through molecular diagnosis, only at the molecular level. Okay, recombinant DNA technologies, PCR, which is polymerized chain reaction, ELISA, enzyme linked immunosorbent assay, are some of the technologies of molecular diagnosis. So there are some very important technologies. Early diagnosis of bacteria and virus in body when the concentration is extremely low can be done by PCR, since it amplifies the DNA several folds. So, as I said earlier, early diagnosis is very necessary as the chances of survival is very high. So, early diagnosis of bacteria and virus, which when they are present in the earlier, the concentration is very low, right? When they grow, concentration becomes high, but at the early stage the concentration of these bacteria virus in the body is very low. So in this time the diagnosis can be done with the help of PCR, as it amplifies the DNA into many folds. Okay, PCR is used to detect HIV virus in suspected AIDS patients and mutations in genes in suspected cancer patients. So, PCR is basically used to detect the HIV virus. As I said earlier, when it is in the low concentration, PCR is used to diagnose the bacteria and virus. PCR is also plays very important roles in the detection of HIV virus in the suspected AIDS patients. What is suspected? Means someone has idea that you may have the AIDS. Okay, when you have idea that she or he might have the AIDS. Okay, this is what PCR. Mutations in genes in suspected cancer patients. So as we all know that cells becomes cancer cells largely because of mutations in their genes. So mutations may affect different genes that control the cell growth and their division, right? So mutations are very harmful. This can be detected by PCR. ELISA is based on antigen-antibody interaction. In the presence of an antigen, the antibody produced against it can be detected. So first of all, you should know the full form of ELISA. ELISA full form is enzyme-linked immunosorbent assay, which is very important question. You have antibodies related to certain infectious conditions. Antibodies are the proteins that your body produces in response to the harmful substances called as antigens. Okay, so in the presence of an antigen, what happens? Antibody is produced which can be detected, and this is what antigen antibody interaction. So ELISA is based on this concept. Then hybridization with a radioactive probe. So this is a very easy approach. In this, the basic gene is hybridized with a radioactive probe, and autoradiography is mainly used here for the detection, and the regions where the mutations occur, large differences are present in the gene that will not be appearing in the photographic film. So the ASIT probe will not capture this type of part. Okay, very simple. In this approach, gene is hybridized with a radioactive probe and autoradiography is used for detection. The regions where mutation is present in the gene will not appear in the photographic film since probe will not be able to find with this part. Very simple approach.